Special session on lipidomics. Pacific Northwest National Lab is one of 17 labs, and one of the capabilities we're known for is mass spec, and that is sort of what's led us down this track of lipidomics and to write this special session, primarily because there are so many challenges when it comes to analyzing this type of data and really taking advantage of the rich, let me put this in action mode. Taking advantage of the richness of this data. So we broke our session into 3 areas where we really thought, there's a lot of very interesting work going on. And one thing I realized when I was looking back over this morning is that it's really an amazing international group of speakers that we have lined up. The first session area is just, the classic lipid identification. It's still a major challenge in how we identify and quantify lipids. The second in pathway inference, how we place these the lipids into biological relevance. And then finally, the sort of systems biology, the complex interaction areas. So I've been working in bioinformatics a long time, but I'm a statistician by training, so I don't try to pretend like I understand the structure of lipids. So for that reason, I have asked our lipids expert from PNNL, Jennifer Kyle, to join me in this session and give a brief introduction to lipids from the perspective of somebody who works on the front line on collecting this data and trying to interpret it. So I see Jennifer just brought her video on, so I will mute myself and let Jennifer take over from here.
Well, thank you, Bobbie-Jo, and thank you everybody for attending this session. As Bobbie-Jo mentioned, we've been working together for quite a while and been diving into lipidomics at the Pacific Northwest National Lab for about 8 years. And so throughout that 8 years, so my role at the lab is managing our lipidomics platform and working with people like Bobbie-Jo and analytical or computational scientists and our software scientists to try and drive this field forward and learn as much as possible and towards understanding the lipidome. So here's our typical lipidomics workflow, which I'm sure many of you are very familiar with. We have total lipid extraction techniques and methods, which are primarily solvent-based. The main ones in which we use were developed in the 1950s. So they're really well established for global lipidomics with a couple new options published a little bit more recently in the 2000s. Our standard platform we use at PNNL is liquid chromatography tandem mass spectrometry, and the development of electrospray ionization has really moved lipidomics forward quite a bit. So, prior to that, we had to separate our lipids using TLC plates, if you guys have had the pleasure of doing that, and then scraping your lipid spots off the plates, breaking them apart into their components, and then looking at the fatty acids on their own using GC mass spec. But with electrospray and the newer technologies that we have, we no longer have to break apart the lipids. We can keep them together such that we know what fatty acids are connected to what type of lipid, like what head group, what type of phospholipid. So we've really made some wonderful advancements in the past 20 or so years in that area. And along with that, we are able to really improve our identification. So again, instead of looking at all the components of the lipids, we're able to look at lipids as a complete molecule. So Lipid Maps has been wonderful resource in Swiss lipids, and they have established a database where, one, they bring together a classification system to try to bring some uniformity in the field, because there's different areas of science have different ways of annotating lipids. So trying to bring together a classification system and an annotation scheme, which a lot of us can use to bring some uniformity in our field. And then, once we have our identification, so again, we use the LC tandem mass spec approach, but you can also use shotgun lipidomics, where you'll know the head group and you'll know the total number of carbons and double bonds. And again, that offers quite a bit of valuable information. So in terms of what we've done for lipidomics, you know, we have these really nice general lipid extraction methods, mass spec methods, which allow us to identify lipids. And also, when we have tandem mass spectrometry, we create an additional avenue to try and identify novel lipids. As lipids fragment in predictable ways, we can again go back into the data and try to identify novel lipids using that information. And then once we have our identifications and our abundance values, there's statistical approaches that we can use to tell us what features may be important within our system if you, let's say you have a control system and an infected system or some sort of disease system. And so this is fairly well established. Again, still quite a bit of work being done to further identify and looking at statistical approaches specifically for lipidomics data. But at the lab, you know, what we kind of run into quite a bit with our sponsors, and because we have a national user facility, and also going to conferences and presenting at conferences and having poster sessions, the common question I get when I give people data is, well, what do we do with it now? That's a very common question. So next slide. There's a couple of steps, and I think this is where this group and the computational scientists can really be helpful in this space, and the people that develop software and working with the biologists. So a couple of different approaches, which this session is going to talk to today, is how do we take this, the lipids and the identifications, and take that information and translate it into understanding the function of lipids a little bit better. And so we have a couple talks that have developed approaches to do enrichment analysis using lipidomics data. And then also, once we have that information, and also how do we use lipidomics data to try and understand what's going on in the lipidome, but also how to integrate that data with other omic platforms to drive our understanding of the mechanisms and, again, the functions that lipids have within a particular system. So we really want to embrace the complexity that lipidomics offers and try to understand as much as possible. And I think there's quite a bit of work being done and a lot of work still to be done, which makes it very exciting. Go to the next slide. So just starting off again with lipidomics characterization. So we have a sample, we identify all the lipids that are possible using our mass spec approaches and also identification software. And then we come up with, we have an abundance values, we do our statistical analysis, and commonly we have these heat maps, which I'm sure many of you have seen these presented in conferences and on posters. And so here's just an example of over 400 lipids identified in human plasma and all the different subclasses in the green which were identified. And then we have our system of comparisons. So these are survivors versus fatality of Ebola virus disease in human patients from Sierra Leone. And so we can see a lot of things are going on in the lipidome, and a lot of things are changing with the function. And so how do we move this information forward into driving what is changing, why is it changing, and how is it changing? Right now, we have a lot of understanding of lipids in general. We know triglycerides are related to energy. We know that, phospholipids have a lot to do with the membrane structure and the fatty acids, the fluidity, signaling lipids. So those fatty acids that are cleaved off and can be oxidized, become signaling molecules. We have some very general knowledge on what's going on there, but I'm sure we can dive deeper into understanding, again, what these lipids are doing and how can we bridge them together with other omic technologies to understand the function. So next slide. So today we have a couple excellent talks. We're going to be talking about two different softwares that are looking at enrichment methods. As I mentioned earlier, Lipid Maps, which came about in the early 2000s, they tried to bring together, they brought together a classification scheme and an annotation scheme. And the nice thing about this is that there's a lot of information just within the annotation of a lipid. So when you parse apart a lipid identification, or a lipid identification, so the first one we have as an example is this PC. So it's a phosphatidylcholine, so that's a subclass of lipid. And you can see off the arrows to the left there, we know it's a PC, so that tells us it's a phospholipid, it's specifically a phosphatidylcholine. And then within the parentheses, we know we have two fatty acids, so it's a diacyl glycerylphosphatidylcholine. The first fatty acid has the 16 carbons and 0 double bonds, meaning it's a saturated fatty acid. And that second fatty acid is has 20 carbons and 4 double bonds, is a polyunsaturated fatty acid. So information, there's a lot of information in the literature on, palmitidic acid and also arachidonic acid. So we can get a little bit more information potentially what's going on there. And then we can also look at the total number of carbons and total number of double bonds, like you do with shotgun lipidomics. But the point of this is that there's a lot of information that's within the lipid name that can help us get to that next level of what potentially is going on in our data. And then how do we use this information that's within the name to come up with statistically significant enrichments to see in our data to try and get us to that next level of what is changing within our system. And then below that is another example using ceramides. So just how can we use the name, the lipid name of what we identified to get us to understand what's going on. So in the next slide, so once we have our lipids, we can kind of understand what's been identified, who's changing, and then parsing it apart using these enrichment tools, potentially what feature or what type of ontology or fatty acid is statistically enriched. How can we bridge that information with other omic information? And so bringing together, metabolomics, proteomics, transcriptomics with our lipidomics really can hopefully, it will bring us to that next level of understanding the mechanisms in which driving those changes in the lipidome. So here's an example here of where I pulled the data from the Lipid Maps protein database. And so I took all the proteins within that database, and for humans, I think it's around 1,500 or 2,000 proteins. And then on top of that, I mapped our proteomics data for this infected system. And so you can see here all these different pathways, such as fatty acid metabolism, glycerolipid metabolism. There's quite a bit going on at the protein level. So how do we bridge together what's going on with these changes in the proteomics to the changes that we see in the lipidomics? And so what tools exist for that? How can we go about doing that? How do we start networking these together? Because it can still be quite a manual process. So this is a great area to really dive into in bridging all these different omics to really understand the system of what's happening. And so lastly, my final slide here, again, I just really want to go into embracing the complexity of the lipidome. And I went to a meeting last summer where I heard a few speakers talk about how do we simplify the lipids. And I really would like to just talk about, there's so much information and, if we embrace this complexity, how can we can really move the field forward. So here's an example here of potentially what happens when you simplify it, or, what is really interesting with these complex systems. So here I mapped using this PathView software, which is a really nice interface. It's freely available, where you can map proteomics data or transcriptomics data, metabolomics data. And then where things kind of fall apart a little bit is when you try to map lipidomics data. So you can see here, the boxes contain the proteomics data. So this is a time series analysis. And then when you try and map the triglycerides, you lose all the information. And so off to the right is a triglyceride profile from our lipidomics experiment. And I apologize for the blurriness of it. But you can see there's certain level of triglycerides that are going down and some that are going up. And so there's two things that are lost here when you try to simplify this data or map it onto these existing pathways. one thing you do is you actually lose the fact that triglycerides are changing. So when I took the average of the triglycerides, the log2 fold change value of what's going up and what's going down, you end up with a neutral change. Like there's zero change going on. So you completely lose the fact that the triglycerides are dynamically changing within the system. And the other thing that is lost when you simplify it, or we don't have really good pathways to work with this data, is that this data, the triglycerides are organized based on the total number of chain carbons and double bonds. And so you can see the triglycerides with shorter chains, fatty acids with less double bonds or that are saturated, they're going down, whereas the triglycerides that contain polyunsaturated fatty acids or long chain fatty acids, very long chain fatty acids are increasing. So by reducing the complexity, one, we neutralize the fact that the triglycerides are changing, and then we also lose this detail of the signature of the chain link having an effect in the directionality of the system. So today we're going to hear some wonderful talks on databases and approaches to identifying lipids and identifying novel lipids, how we can use these identifications to for enrichment analyses of lipidomics, and then also integrating our omics data together to really understand the mechanisms that's driving these changes in the lipidome and how the complexity of the lipidome, although it can be a little daunting, believe it has a lot of potential to drive biology forward.
Thank you, Jennifer. That was a great introduction. I'm going to stop sharing here. So just as a reminder of how this will go, I am your moderator. And we have 2 minutes at 3:40. The first presentation by Maria Fedorova, or she'll tell you how to say her name appropriately,